Everyone, and this is a lecture-style video on laboratory safety. There's not going to be too many lecture-style videos in this class for me. It's mostly going to be technique videos. There is a playlist up on YouTube, which this is actually a part of, oddly enough, but if you're getting this through a link and not through the YouTube channel, there is a playlist on YouTube with all of our safety videos, protocol videos, and the like. So this is our laboratory safety for biology 2420 and 2421. The goals of this is to describe the use of PPE, describe and adhere to BL2 safety protocols. This also has our labeling protocols in it, as well as in a different PowerPoint on a different video from earlier today or later from today. Earlier from today, I recorded it later today. I recorded it earlier today. It'll be up on the YouTube later today. Emergencies video. Now, SMTE 0092 is an online Blackboard course required for all labs. You are required to take it. It's still required for online learning and complete it as soon as possible. The deadline for extra credit for this course for assignment 0 is the 29th of January 2021. I believe that you have until the end of that week, until it's Sunday, which would be the 31st of January, to complete it, or the IT and EH&S start removing you from the course. I do encourage you to follow along on this PowerPoint. This PowerPoint is up on Blackboard. So we're going to start with some personal protective equipment, or PPE, used in a BL2 lab. The first big one we have are gloves. Lab coat, shoes, pants, safety glasses, hot gloves, and scrubs. You don't use all of these all at the same time, but they are necessary. Personal protective equipment is gear that you don or put on to protect yourself from pathogens, chemicals that might be in the lab, or some other type of safety issue that may arise in the lab. Gloves go on your hands. As you can see, I have a set of gloves in that picture as well. Lab coat goes over you. There is a very short video on YouTube on how to properly put on a lab coat. There's also a video on how to don and doff gloves. You do want to have closed-toed shoes. If you do not have closed-toed shoes, you would not be allowed in the lab. Safety glasses, if necessary. I have a set right up here. Let me pull them down. Nice set. These go over my actual glasses, like so. Pants are also a type of PPE. You are to have long pants in this lab. If you do not have long pants, or you have shorts, jeggings, the like, you will be asked to leave. You also need to have hot gloves, which we'll go over what they are. They're for things like autoclaving and other hot things, boiling water and the like. Scrubs can also replace either a lab coat or pants. Let's start with your lab coat. Your lab coat is an external covering. It is a physical barrier between the chemical or the organism and you. Color isn't really that important. I have two navy blue lab coats, mostly because I like navy blue. Most of the lab coats you're going to get are going to be white. The reason you want white lab coats, usually, is because the white lab coat, when it's over you, you're able to determine exactly what was spilled on you if it has a color. The length of the lab coat is also important. You want it to at least go down to your thighs, if not down further past your knees. The longer it is, the more protective it is. So the goal of a lab coat, as I said, is to protect you. So the longer it is, the more protective it is. Safety glasses or goggles are the eye coverings, which I have right here. And when they go over your eyes, they are to prevent aerosols or water from entering. Right, like that. And then let's that over like that. And as you can see, it protects my glasses, it protects my eyes, and it does create a seal that prevents any type of, well, most types of contaminants from getting in and damaging my eyes. You need to use goggles whenever you're using a vortex or any means of creating an aerosol. If you're going to be creating an aerosol, you need to use the safety glasses or a biological safety cabinet. Gloves act as a barrier. In a clinical setting, they prevent cross-contamination. There is a video, which I will have linked here, proper removal, as well as on Blackboard, of how to properly put on or don and take off or doff your gloves. Hot or autoclave gloves are insulated gloves that prevent heat transfer, and they're mostly so you don't get burned. Pants are a leg covering, and typically it needs to be of a thick material. Denim, fabric, khaki, the like. No jeggings. Any thin material that you have will be asked to leave. If you do not have long pants, you'll be forced to leave. It doesn't matter what your gender is, doesn't matter what your sex is. Long pants of a thick material are necessary for the lab. Closed-toed shoes are also necessary. No Crocs, no sandals, no flip-flops. Closed-toed shoes only. If you don't have those, you'll be asked to leave the lab. Scrubs are coverings for the feet, body, and legs. They're typically used in hospitals by nurses or doctors, and we do allow them for BL2. Do keep in mind that all PPE that you have that needs to be brought into the lab stays here. So lab coats, scrubs, lower scrubs. Gloves are disposable, but goggles, safety glasses are brought into the lab, and they stay here until such a time as we are able to disinfect them. Now, let's talk about some basic protocols. Donning and doffing your equipment, your entrance procedure. The first thing you do when you walk in is make sure your hair is up. I have relatively short hair, but if you have long hair that's down to your shoulders, be sure it's up in a bun away from your face. This is a safety hazard. If you have facial hair like I do, be sure that that's either short enough so it won't get caught on fire, or you have some kind of covering. We don't want you to set your beard on fire. We don't want you to set your hair on fire. That needs to be done before you enter the lab. Before you enter the lab, make sure that you have closed-toed shoes on. You have long pants on unless you're putting on scrubs. You then want to come in and place your book, pencil, and Sharpie at your workstation. Wash your hands. We have 4 hand washing stations in each lab. You then want to don or put on your lab coat. This is going to be a link as well. It's also on Blackboard. We need at least 2 buttons per our safety protocols, but we recommend as many as possible. My lab coats, one of them has 7 buttons, one of them has 6. I usually do up all of them. You then want to don your gloves and get started to work. When you're leaving or you're doffing your equipment, you want to doff your lab coat and store it, properly doff your gloves, wash your hands, and then exit the lab through the designated exit area. You need to don your equipment every time you enter the lab. You need to doff your equipment every time you leave the lab. Your equipment stays in the lab. Long hair must be kept above the lab coat collar, and this is any length of hair that goes below the lab coat collar, regardless of sex, needs to be put up. If that means that you put it in a nice bun or a man bun before you come in, so be it. That is the protocol. If you have a long beard, the beard will have to be tamed. What do I mean by that? I mean it has to be put up as close to your neck, your throat, and your face as possible. And if that means you wear something over your beard to make sure that you don't catch your beard on fire, so be it. This is a fire hazard. We do not want you catching your face or your head on fire. Disinfection. Lab bench should be disinfected when you arrive after donning your PPE and should be disinfected when you finish before doffing your PPE. Assume that your previous lab person did not disinfect it. Again, there will be a link here. I just have to hyperlink it. It's on YouTube of a disinfection demo where I disinfect a lab bench for you. Pipetting should always be done with a pump or a bulb. No mouth pipetting. I don't care if you learned it in high school. I don't care if you know how to do it. No mouth pipetting. Glassware should be washed with soap and water after use. Glassware used for culturing should be autoclaved. Autoclaving is a method of steam sterilizing equipment. You'll hear autoclaving a lot. this semester. When we're talking about our glassware, we talk about autoclaving the glassware. When we're talking about our lab coats, we autoclave the lab coats. An autoclave uses extreme heat and pressure and steam to sterilize, not sanitize, but sterilize some form of equipment. Some plastics, most notably Nalgene, are autoclave safe, but most of them are not. Glass is autoclave safe, as is most high-grade lab coats. If you have a lab coat that is a lower grade or is a disposable lab coat, it will not survive an autoclave. If this is the case, speak with your TA or me, and we'll see about getting you a lab coat that can be used multiple times and autoclave multiple times if necessary. Fire is an integral part of disinfection. We do use Bunsen burners, and I go over this more specifically in our emergencies video, but this is a fire hazard. Hair is a fire hazard, as are the Bunsen burners themselves. We do use Bacti incinerators as well, but you may be required to use a Bunsen burner in the lab. If there is a spill, inform your instructor, TA, or supervisor if one of those were to happen. In our lab, only instructors, TAs, and myself can clean a spill, but we do provide a demo for you just in case you ever need to. Broken glass and other sharps should never be picked up with your fingers. You should use tongs or tweezers for a bench top. You should use a broom and dustpan for the ground. Place broken glass in sharps containers. There are 2 sharps containers in each of the lab. There is a big one and a small one. They both are cardboard with a red biohazard stamp on them. Those are for contaminated biohazardous glassware. If you don't know that it's biohazardous, it's biohazardous. The assumption is it's biohazardous. If you know for a fact something is not biohazardous, we do have a floor model standing sharps bin for broken glass in the micro prep room that is attached to both of our labs. Some basic labeling. All cultures are to be labeled at the time of you making them. That is before you inoculate them. This is done on the bottom of the plate, vertically just below the cap of a test tube. There is a linked demo as well. Most labs, depending on where you are, will have their own requirements. Are your initials or name, class number, section, media type, the inoculum in genus and species or an abbreviation, the date, and incubation temperature. In the video here, I used mannitol salt auger and Staphylococcus aureus. For our pictures, we have this one here is unknown number 1, TSA, 37 degrees, 421, the initials of the person who's doing it, and the section. These are what you would most likely be working with. Your unknowns, if you have an unknown for 2421 or the bacteria for 2420, the media type right here, incubation temperature, date of inoculation, your initial, and your section. Your section is important because we store our plates and tubes by section. This is one of our stock plates. This is a stock Bacillus megaterium plate right before I inoculated it. And as you can see, I have the genus and species as well as the other information necessary. For tubes, this on the left is what a tube that you would use looks like. And for our stock cultures, we do have labels that we use instead. Now, disposal and tips. Sharps are disposed in their personal receptacle. Inoculated media is disposed of on the floor biohazard bins. Slides, pipette tips, Kim wipes, things that you would use at the benchtop are disposed of in the benchtop biohazard bins, and your gloves go in the biohazard bin. It is a massive safety violation if the gloves end up in the trash can. And finally, some best practices and tips. You want to avoid touching your face in the lab. Your fingers, once they go near your eyes or your nose or your mouth, they have access to your bloodstream from there. You always want to use a rack when moving cultures to prevent spills. Do not walk with hot laboratory utensils. We don't want to have a needle on fire just walking through like a torch. You want to familiarize yourself with the exits, fire equipment, shutoff valves, and other emergency equipment in the lab. This also includes things like SDSs, or safety data sheets. Safety data sheets are provided to you in the lab, as well as our biosafety protocols that are approved by the Institutional Biosafety Committee, as well as our exit procedures in the case of an emergency. The last of those, there is a specific video on exit procedures on YouTube waiting for you to watch. And finally, no mobile devices in the lab. You'll notice that a lot of our pictures that we have are using mobile devices. That's because the mobile devices have a better better photo quality than the webcam that I'm talking through to you now. This is an exception. This is not a normal thing that would happen in this lab. In labs, no mobile devices. And the reason for that, I'm going to hold up my phone here. This is a Google Pixel 3a. It's a rather large device that goes right up to my ear, or I flip through. If bacteria is on here, it's now on your face, down your face, other side of your face, all over your hands. And you can really spread bacteria if you have mobile devices out. So no mobile devices in lab. It's not just a be polite to your TA thing. It's also a safety hazard.